My name is Sylvie Le Guyader, and I'm the director of the Live Cell Imaging Core Facility at Karolinska Institutet in Sweden. In this video series, we are talking about assessing the imaging efficiency and the risk for bleed-through for your sample and on your microscope. This is the second part of 3 videos. In the first video, you've learned the basics of fluorescence microscopy, and in this video, you're going to learn how to know your sample and your microscope better. First, I would like you to pause the video and answer the 2 questions below. This is not a test. At the end of the video, I will ask you the same questions again. This is a way for you to summarize for yourself what you know already, and then you watch the video, you learn some things, and then we summarize again what you have learned. How do you know for sure which fluorophores are in your sample? And where can you find out which lasers, filters, and dichroic mirrors are available on your microscope? Okay, so write down these answers and then start the video again. The first thing we're going to do is we are going to find the exact names for your fluorophores in your sample. GFP is a large family of green fluorescent proteins. And if you go to fpbase.org, this website, you will see the whole family tree. GFP was first isolated from a jellyfish, Icoria victoria. So the first GFP that existed was called AV GFP. But this GFP was very weak and it was bleaching very fast. So it was quite quickly evolved to another GFP called enhanced GFP, eGFP. And this was brighter, but it was a weak dimer, which created problems. So then this GFP in turn was evolved into a true monomer, mEGFP. However, GFP is written in many articles regardless of which green fluorescent protein was used, and that leads to irreproducibility. So which fluorophore do you really use? Another example is the rainbow and confetti mice. In the original articles, it is written that the authors used CFP, GFP, YFP, and RFP. But there is no fluorescent protein called just RFP, red fluorescent protein. This is also a family of protein. So which fluorophores were really used? Here are the emission curves of a few of the fluorescent proteins which have RFP in their names. As you can see, there is a wide variety. And this variability is especially true in the red, far red, and near infrared range of fluorophores, because this is where most of the development is happening. Fight irreproducibility in science. Check the exact names of the fluorophores in your sample. And you can do this by looking at the original tube, if you have bought a plasmid or you have bought a dye, for example. You can look at the product data sheet that you can usually download from the website of the vendor. You can look at the original article that reported this dye or this fluorescent protein, and you can sequence it and then search in FPBase. You go to Tools, FPBlast, you enter the sequence, and you know exactly which protein you have in your plasmid, and this is the name you write in your article. Now you're going to pause this video and download a form from our website. The link is below the video. So this is what the form looks like, and what you will do is that you will write the exact names of your fluorophores on row number 2 here. Once you have done that, start the video again. Now that you found the exact names of the fluorophores in your sample, the second thing that you need to do is find the full spectra, not just the peak, for the fluorophore. The excitation and emission peaks give no information about the shape of the spectra. Always use the full spectra to evaluate bleed-through, not just the peaks. So this is an example of DAPI, So the excitation spectrum is the dashed line, and the full line is the emission spectrum. So what we can see is the peak of excitation for DAPI is 367, and the emission peak is 461 nanometers. One thing that you want to do when you see this type of spectra where the curve is truncated is that you want to fill it in, because this just means that the person who has measured the spectrum has stopped measuring, but clearly the spectrum would keep on going. What you can see in here is that DAPI emits a majority of its photon in the blue range. It also emits many photons in the green and in the red range. About a third of the photons that DAPI emits are not blue. You can also see that the 405 laser that we usually use to excite DAPI on laser-based systems is very far away from the peak. But even if we excite that at a few percent, we anyway see it because it is so bright. Here are some more examples of what the peaks do not tell you. In this graph, you can see the excitation and emission curves of DS-red and the excitation and emission curves of M-cherry, two red fluorescent proteins. What you cannot see if you only get the peaks is that M-cherry is really excited in the UV. And DS-red has two big excitation shoulders where the fluorophore is actually very well excited. Also, M-cherry, the emission is really red shifted compared to DS-red. So how red is the red fluorophore in your sample? There are very large variations in fluorophore spectra, especially in the red, far red, and near infrared range. To find the full excitation and emission spectra of your fluorophores, where can you do that? You can go to the datasheet of the fluorophore or the dye or the antibody that you have purchased. If it does not show the full excitation and emission spectrum of the fluorophore, ask the company, they must give it to you. You can also go to online spectra viewers like fpbase.org, Thermo Fisher, Semrock, all have excellent spectra viewers that you can use. If you do not find the spectra of the fluorophores in these spectra viewers, you can also do an online search for the name of the fluorophore and spectra. But Google will always find things to give you, so make sure that where you are looking at is indeed correct. And you can go to the original article describing the fluorophore. So work with the full spectra, not just with the peaks, and write to suppliers and demand the full spectra. It is not possible to evaluate bleed-through if you do not have the full spectra, to tell them that they have to give it to you. So now you're going to do a little exercise. You're going to find the full spectra of your fluorophores, including any nuclear dye, and then you will write in the bleed-through evaluation form that you have downloaded at which wavelength the excitation starts and peaks and ends, okay? So here we have written the name of our fluorophores, and in here I wrote the start and peak and end for EGFP. And you can do this both for the excitation, so this is the excitation, and for the emission, you would write down where it starts, peaks, and ends. Do that, and come back to the video afterwards. So now it's time to practice. So pause the video now and answer the quiz below, and then start the video again. Why is it important to find the full spectra of fluorophores, even if the company that sells them only writes the excitation and emission peaks? So write your answers and then come back to the video. The reason why it's important to work with the full spectra is because the peaks only show the best parts of the excitation and emission curves. The full spectra also show all the troublesome parts like shoulders, bumps, and widely falling curves, which are mostly responsible for cross-excitation and bleed-through. So it is not possible to evaluate cross-excitation and bleed-through without the full spectra. I'm going to work on finding the specifications of your microscope. So where can you find all the lasers, filters, and dichroic mirrors in your microscope? Most of the time you can find them in the software. So open the software and find all the possible excitation sources and the dichroic mirrors and the filters. So don't hesitate to ask for the person in charge. Have a look at the facility website. Sometimes it's written there as well. You can also ask the company that sold the microscope to you and refuse meaningless answers like, this is the FITS filter. What we need is numbers. We want to know exactly which wavelengths you have in your microscope. So once you have everything, you're going to write it down in the form. Lasers, or maybe they are filters if you have a system that works with LEDs or lamps. So let's say that your excitation filter is 350. Right from here, it starts at 325 and it ends at 375, so plus minus 25. Okay, so you're going to do this for all of them. If you have a laser, you just write the laser here, and then here it's just one wavelength. And then you're going to find all the possible emission filters. So you can have fixed filters on your system, and in that case it's very easy, it's the same thing. So you're going to write the emission filter, and then you will write again where it starts and ends. In our case, we have filter-based systems. For example, for DAPI, this is the excitation filter we have, that's the dichroic mirror, and this is the emission filter. So I would write in here 460/50, and here I would write 460 minus 25 and plus 25. Your system might be different and might use spectral detectors. Many point confocals use spectral detectors. How do you know that your system uses a spectral detector? This is if you can change the width of the emission filters. For example, on a Zeiss confocal, it may look like this, where you could slide these to make the filter wider or narrower. In this case, you're going to find in the software what the filter is set to. So write down in the form the specifications of the filters as you normally set them when you image your sample. And then you need to find also all the possible dichroic mirrors. Sometimes the dichroic mirrors are abbreviated BS for beam splitter, but it's the same thing. Ask the person in charge for help. So pause the video now, fill in the spreadsheet as above, and then start the video again. Well done! You have now found the exact names of your fluorophores, you have found the spectra for them, and you have found all the specifications for your microscope. So you are ready to evaluate the risk for bleed-through in your sample on your microscope. But first I would like you to pause the video again and read the answers that you gave to these two questions. How do you know for sure which fluorophores are in your sample? And where can you find out which lasers, filters, and dichroic mirrors are available on your microscope? So what did you answer correctly before watching the video? And now that you have watched the video, how could you improve your answers? So please try to improve your answers and then start the video again. Well done! You have learned the following in this video. The literature is full of confusion about which fluorophores are used. This is a source of irreproducibility in science. So please, when writing an article, check the exact names of the fluorophores in your sample. And you can do this by sequencing your plasmids and doing a blast search in FPbase, for example. Or you can look at the dye's original tube. Make sure you write the correct names in your article. You have also learned that the excitation and emission peaks do not provide enough information to evaluate cross-excitation and bleed-through. One must look at the full spectrum. And then you have hopefully being able to build a list of all the excitation and all the emission wavelengths, as well as all the dichroic mirrors available on your microscope. But how would you use these tools to evaluate if there is a risk for bleed-through when imaging your sample with your microscope? So watch the next video to find out. This is part 3, assessment of the imaging efficiency and risk for bleed-through.